Hello and welcome everyone to today's webinar. My name is Jennifer Caveney and I will be facilitating today's presentation. We have two excellent speakers today that I would like to introduce. Our first speaker is Ed Collins, Director of Product and Process Development for the National Food Lab. Ed has a bachelor's degree in agricultural engineering and a master's in food science and has worked in the food industry for almost 35 years. Ed works directly with clients to develop new products, create new processes, and launch new ingredients and bring products to market. During his career, he has worked on just about every form of beverage, nutritional product, meal, or snack, as his team creates new products for clients at all stages of the development cycle. Our second speaker today is Malcond Valladares, and he is a food scientist with the National Food Lab. He specializes in food safety and quality. With expertise in microbial risk assessments and emerging natural antimicrobials, he helps optimize ingredient selection, processing, and product stability. A member of IFT and IAFP, Malcond holds a PhD in food science from the University of Tennessee, where he researched thermal and activation kinetics in foodborne bacteria. All right, with that being said, Ed, I will hand it over to you.
Thanks, Jen. You covered a little bit about who we are here at the National Food Lab. We are an end-to-end innovation partner, right? We work with everybody in the food, beverage, and the supplement areas. We'll do everything from ingredient, technology and new applications, and ideation, co-creation with consumers and consumer vetting of concepts. The two companies here, so Ithaca, New York, and Lafayette, Colorado is a dedicated pilot plant facility, with Ithaca, New York being the larger product development center. So along with product development and feasibility, obviously a lot of process design, everything from beverages to retort processes to evaluation of, I guess, alternative preservation methods. We've done work with high pressure processing, microwave processing, a few others along the way. And we also support product lifecycle management tools and data management support for larger companies that may be integrating new businesses or just trying to understand what types of ingredients and risks they may have in the background of their quality program. Of course, we're part of the Eurofins network, so everything we do as far as chemistry is really world class in that aspect, and we'll pull all that together for you as we work through projects. When you look at the product life cycle, right? I mentioned the product innovation piece, so trend mapping, concept development, rapid prototyping. There'll be an example of some of the trends and some of the types of products that we make, and we'll talk about those. On the product development side, it's really what's the technical feasibility, how can you go about improving something, how do you get more protein, how do you eliminate hardening, or how does this new fiber or plant protein versus dairy protein work in your product. We'll then take that to the pilot plant, everything from process design and optimization to scale up and readiness. We have the ability to bring in unique equipment if there are specific things that you need to test. And then we'll scale that up with you, going to the co-man, doing the process validation, making sure they're meeting specifications, understanding how that plant-produced material performs over shelf life. And then we get into data management, so PLM and spec development. We've done a lot of work in product vision, SAP, a couple of custom systems. We've done spec right as well. So quite a bit of experience in that space. And then what's the project management that goes along with it, right? So as you get into bigger projects here at Eurofins, it may be, how do we include sensory and consumer research? What are we doing for brand development? Are we helping with packaging or processing? What types of things do we need to consider? We'll manage all of that for you as well. So as we get into it today, we're going to focus on some of the R&D tools and strategies for improving products. We're going to talk mostly about water activity control. I think Malcond will cover some of the other regulatory aspects as we talk about those. Really, you're going to hear a little bit about food safety and how some of those things impact product quality and what are the things you need to do to help manage them. So we'll look at mitigation strategies, we'll look at pH and AW control, and then we'll get into the impact of some of the specific ingredients that you might use and how they impact shelf life.
Excellent, thank you, Ed. I will take over from here. And first of all, it's important for us to know where is the space we're going to be playing. And specifically for this type of products, and when I'm talking to water activity control foods, our products are below 0.85. And 0.85 becomes the magical number for water activity control foods, because below that is where we start preventing the growth of pathogenic bacteria. So that becomes essential for the safety of this type of products. And if you follow along, this chart is a very handy tool to define where is this regulatory space for the specific product you're going to have and what type of regulations and laws will apply along the way. So for the purpose of this presentation, we're going to focus our talk on, like I mentioned, water activity control foods. Specifically, we're going to dive a little more into detail on bars. So water activity control foods, and what are the challenges that come along with this type of products in terms of physical and microbial challenges. So first of all, people we will talk about water activity along this whole presentation, but what is water activity? What is the definition of water activity? In essence, water activity is a ratio between the water pressure of the product once it's in undisturbed and in equilibrium with the surroundings, compared to the water pressure of distilled water under identical conditions. So basically, a water activity of 0.8 means the vapor pressure is 80% of that of pure water. So low water activity foods are naturally low in moisture because they even have water removed through a drying process, for example, raisins, and the water activity is naturally low due to the drying process. In terms of water activity control foods, this is a step even further, where the activity of the water has been intentionally managed to prevent microbial growth and extend the shelf life. For example, jams, protein bars, and crackers. So how water activity relates to food safety? Like I mentioned before, the primordial, the most important number here is 0.85. Below 0.85, basically, we're going to prevent microbial growth for bacteria, but it's important to highlight that yeast and mold will still can grow and have an impact on the quality and shelf life of your product. So one step further, if you lower the water activity below 0.6, is when you start inhibiting mold, yeast and mold. And this does not eliminate the pathogenic risk. If bacteria were already in the product, they're just going to maintain stasis, which means that they're not going to grow, but they will be present. However, bacteria will not continue to grow and proliferate in the product. So pathogen persistence is also something we need to take into account once we have post-processing contamination, meaning that if the product has been opened and you have other chances of contamination, what other measures are in place in order to prevent microbial contamination to continue proliferating and further damaging the quality and safety of the product. In this case, we have tools like pH and water activity, which are the only two factors that have been that are enforced by FDA and have their specific guidelines in terms of combinations that can help to prevent and enhance the food safety of a product. So what's beyond the microbial safety of product? How water activity relates to other aspects of food? Water activity can also help to control the rate of reactions of the different activities or the different aspects of food quality in terms of lipid oxidation, rancidity, and browning. So the role of food formulators is basically control this rate of oxidation and lipid oxidation and all those different activities by reducing the water activity through different strategies, through formulation and different ingredients that can be used to enhance the safety and the stability of the product. Browning reactions are related to, for example, the relations between protein and sugar, reducing sugar. And in free water, basically the sugar will reduce, and the reaction will reach a peak at around 0.6, but once you start decreasing the water activity, the rate of reaction will start slowing down. Some useful ways to use water activity in our advantage is use temperature and humidity to increase that reaction rate and use it to understand and extrapolate how these reactions will behave over the shelf life of a product. So you can use this to use and model the shelf life in accelerated conditions. So that's one useful tool we can use to model this type of reactions throughout the shelf life. Okay.
So we'll jump into a case study. We'll look a little bit about, oops, excuse me, one too many. We'll look about a product improvement process that we executed here. Company came to us, existing drinkable medical food, multi-use container, right? So once you start talking about multi-use container, kind of the way you have to store it, the way you have to control it, the way you have to formulate changes. They were at a very high protein level, 17 grams of hydrolyzed gelatin in a 30 ml serving. They want it to be shelf stable. And if we just kind of look at what that entire ingredient line was, the basics of it are collagen, water acid, some amino acids to help with the product's primary functionality, and then preservatives, right? So potassium sorbate, sodium benzoate, and then sucralose. In a product like this, a lot of sugar actually, will increase the likelihood of growth in some cases. So these sucralose is a sweetener. And the client wanted to look at two new versions. One is just an alternate collagen supplier. Can we get a better flavored collagen? Do we want to be, how do we maintain 17 grams? And then the second one was, let's get to a PDCASA 1, also at 17 grams, and what's that product going to look like and taste like, and what type of ingredients should be used? So really the first thing is what do we need to measure? What's important in this product? How are we going to evaluate it, right? So we looked just at what because it's a multi-serve container, you have the risk of incidental contamination, right? So we've got a pH of 4.4, which is good, right? That eliminates a lot of the bacteria to the right of this chart. Our hurdle, though, is that a water activity of 0.97, even at a pH of 4.4, allows for the potential growth of pathogenic bacteria, right? And so when you look at that, you have to understand, is the preservative level going to be sufficient to prevent that incidental contamination? So when you look at the product and somebody's pouring something and they wipe off the rim because there's a drip or whatever, if they contaminate the product, is anything going to grow, and is anybody going to be at risk? As a medical food, this one actually is more important because you are giving this product to an at-risk population, right? So they're under some sort of care. And then does it need a thermal process? How do you make it stable in the first place? What does it look like? What are you going to do with it once it's done? Right. So obviously the water activity is too high. We got to get it below 0.85 to help eliminate the pathogenic risk, right? I talked a little bit about how do we manage the incidental contamination, and then what's that challenge study looks like. So what does that do to the product, right? So if we're not going to use, say, reducing sugars and put a lot of sugar in that, glycerin becomes a primary humectant. It'll bind the water, it'll minimize the water activity. Now, what it'll also do is it will outcompete your ingredients for the water. So when you look at the likelihood, or like different ingredients will compete for that free water in the system, glycerin's going to outcompete everybody. So that will now drive or change the solubility of the collagen that's already in the product at the 17 gram formulation, or even the protein that we're trying to put in to manage, to get to that PDCASA1, right? And so when you get glycerin in there, what happens, right? At 17 grams, all the dairy proteins start to drop out, right? So even the hydrolyzed proteins drop out. It's just not practical to have that level of protein in the system, so you have to drop it. You know, it ended up being a 12 gram where you got the solubility and it stayed clear and product didn't precipitate. Now for the 17 gram product, the glycerin also required that we go to a lower, a smaller fraction of hydrolyzed collagen. So we had two new options at 3,000 and 5,000. The 3,000 helped manage the viscosity and the product quality. Now, the problem is the glycerin and the hydrolyzed proteins, whether it's collagen or whether it's a dairy protein, are going to be bitter. And so as we looked at that, there becomes an increasing need for flavor systems or taste modifiers to help hide that bitter impact in both those systems. In fact, for the dairy protein, it even got to going from citric to, say, malic acid to change the flavor profile and hide some of those notes using a different flavor system. And then, fortunately, with the sorbate and the benzoate pH water activity control, that product would now pass that incidental microbial challenge test, and the product is really safe at this point for the intended application.
Excellent. So now let's dive into the protein bar stability piece of this conversation. So a quick overview in terms of what the market trends are right now. People and consumers are always demanding for higher protein snacks. There's trends such as keto, Quest chips, where the challenge is to try to fit more and more protein into the same serving size. So the overall protein bar global market is on the rise. If you can see the graph on the right side of the screen, it's projected to keep increasing over the next couple of years. So in general, protein bars face some challenges in terms of texture, flavor, and those challenges are exacerbated when you start increasing the amount of protein in the system. So in terms of what strategies and what can we do, we can manage this through ingredient technologies. We can make products that are shelf stable and through different ingredient application or different order of validation and different strategies, we can manage these challenges in terms of hardening, migration of moisture, and overall increase the product stability. So some of the formulations approaches also include ingredient selection, what type of milk proteins or what type of plant-based proteins are you going to be adding to your protein bar? What's the role of carbohydrates and how these type of ingredients can either enhance or decrease the amount of hardening you will have in your base product? What is the importance of overall ingredient selection in the shelf life stability and product development activities that you're going to follow? So in general terms, there are a few key ingredients in protein bars. Mostly there are composed of protein, either dairy or plant protein, and a sweetener systems. So the little table here, you can see a breakdown of different type of categories of protein bars. You have the more specialized that are for sports nutrition, which tend to have a slightly higher amount of protein. Keto bars, which are high in fat, and you have like the breakfast bars, which are a little more loaded in carbohydrates because they're meant to give you energy at the beginning of the day, and snack bars. So, but overall, the key element here is that the water activity needs to be below 0.6. Some of them you'll find that are products that are still above a little 0.6, but there's other strategies that you can implement to control the potential microbial growth. So the type and proportion of the ingredients is important because it has a direct impact on stability. And stability means hardening, color change, Maillard reaction, and all the detrimental effects that you can have in hindering the shelf life stability of the product. So what are those key elements in the shelf life of a protein bar? Basically, consumer acceptance, flavor and taste is king. So if consumers are not going to accept the product will have a shorter shelf life, basically. And so this is how you end up establishing what is your expiration date or best by date, what's the amount of time that you can have and prolong the shelf life of your product and continue to keep the organoleptic attributes of your product intact. Also, there's consistency challenges when you start formulating products and you do the scale up. You need to make sure that you can produce the same product over and over in a consistent manner, including no off stays, no mold growth, textural changes are tried to be minimized despite having the same recipe, right? And also, what other elements in terms of microbial spoilage? Most of the time, with products that have low water activity or water activity control foods, you're going to be dealing with molds, yeast and molds, specifically xerophilic molds that can still grow below 0.6. They are rare, but they still can be present, and it can still be a problem for this type of products. And also, you have chemical reactions that can drive sensory, meaning browning or lipid oxidation that can cause negative sensory changes that can render the product unacceptable. And from the physical point of view, changes that occur during the shelf life of protein bars are mainly excess of protein can cause case hardening or bar hardening over time, and also lead for product reaction at the end. So it's important to specify or establish what are the specifications for release of your product in terms of what is the water activity you're going to be measuring, what is the release criteria, what is the minimum or maximum water activity that you're going to have before releasing that product, what is the packaging you're going to use. Is it going to have a packaging that is a moisture barrier? What is going to be the storage and distribution? Is this product going to be stored in a warehouse that is hot in Arizona during summertime, or is this going to stay higher up in winter, right, when the temperatures are going to be lowered? So in terms of physical stability, most of them are going to be related to texture, visual, and sensory changes. Protein and protein interaction can cause hardening, and mild browning are undesirable. During storage, protein will start interacting and cause basically disulfide bonds that are going to start creating protein aggregation, and this is going to lead to phase separation, which is also going to lead to protein hardening. And you can see that in the next slide, where you can actually, the illustration can show how those proteins are aggregating over time, and can also lead to protein glycation when the protein is actually interacting with the sugar, and it can cause hardening over time. So this is led by multiple mechanisms, and it's exacerbated by temperature, so it's a thermodynamic driven type of reaction, and are different influencing factors that can also help to increase the rate of reaction, meaning the pH of the product, the ionic composition, storage temperature, and concentration of protein, because the more protein you have in your system, the faster this type of reactions will occur. So in terms of what strategies you can use for milk proteins, as an example, this study shows the combinations and blends of protein can actually have a beneficial effect over time. So when you start mixing proteins, meaning whey protein isolate and whey protein hydrolysates, they tend to render the protein softer over time, so they due to a synergistic effect. And so combinations over single proteins are preferable for overall protein softness over time. And in terms of carbohydrates, the type of carbohydrate also plays an essential role in terms of hardness. Different carbohydrates can render the protein harder. For example, fructose can create one of the hardest bars, and also can be considered reducing sugar, which will drive the browning over time of the bar, especially when you have bars that are colorless, like vanilla flavor. Those are going to be more noticeable. In the image on the bottom of the slide, you can see how different sugars would interact with different type of whey proteins, sodium caseinate, caseinate hydrolase, and how over time they change and they start causing browning due to Maillard reaction. So in general terms, higher concentration of syrup also reduce higher water activity, but could be a balancing act where you don't want to lower the water activity. You want to lower your water activity. You don't want to create issues with browning at the same time. So what are the strategies we can implement? Basically is set your specifications, measure what are the moisture and water activity of your incoming ingredients. Basically, since you're dealing with cold form bars, it's essential to control the moisture and water activity of the incoming ingredients because there's no further processes that can eliminate water from the system. So matching the water activity of the components that have similar moisture and water activity components is essential to prevent moisture migration in the bars. You don't want to have things that are incompatible mixed together, like nut pieces, soy crisps, for example, fruit, dehydrated food. And also another strategy is to use edible barriers in the form of enrobing, fat enrobing, or chocolate enrobing to prevent moisture migration overall. Blending proteins is also another good strategy. Hydrolyzed protein tend to be softer, but can also become sticky over time. So having blends with other types of protein will help you to manage texture over time. Sugar ratios is important also. Remember that browning can be an issue, so reducing sugar and protein interaction is another thing that you need to manage over time. And setting the shelf life of your product and creating isotherms is also going to be another essential tool that will help you to predict how the product will behave over time, and if there's any opportunities for prevent moisture migration and prevent hardening over time. And conducting an accelerated shelf life or even a real-time shelf life is going to be essential for understanding how these systems will behave and what strategies can you implement to manage the shelf stability of your product.
Thank you, Mal. So in this next one, we'll talk a little bit about managing, well, obviously we're talking about managing water activity, that should be kind of a given now, but also what some of the mixing and some of the processing and incoming raw materials can do, right? So especially bar manufacturer comes to us, they want to talk about some mold growth they're seeing really around the world. So they're shipping product from their location in different countries, and they're getting reports of mold coming back from other parts of the world and parts of the US as well, right? So we started out, let's go out, we'll audit, we'll see what they have, what type of raw material specifications do they have, what are their raw materials that they're using, and then, you know, doing an initial look at that water activity. And with the products that were spoiled, the APC was good, less than 1,000 CFU, 10 to the third is kind of the ready to eat standard. The low yeast and molds were good. You know, the typical ones, 10 or fewer CFU, so that all looks good. But it was, in fact, high for osmophilic yeast. Those were typed. And there's an opportunity there with some of the yeast and the mold. And we've found two types of organisms that could grow in the product, in some of the moldy products. So what are we going to look at? We started by breaking down the product portfolio, right? So 11 bars in the portfolio, measuring the water activity across all of them. As Malcond pointed out earlier, you really want to be below 0.7. In a perfect world, you're like well below 0.6, and you don't have as much of a concern about anything. But realistically, you know, with the types of ingredients they were using, what their target market and who they, you know, the consumers were that they wanted, you've got to have some level of control or some level of quality with those ingredients to meet those that kind of market niche. And as you can see, there's 4 products on there that are over 0.7 that would certainly be a concern for growth. And so we said, what are the ingredients? What are we looking at? How are they really trying to control it? You know, they have raisins and dates into different bars. You know, the water activity, those are great. You know, 0.536, 0.56, well below the target. But some issues potentially with the brown rice syrup they were using, which which has a much higher water activity. As you can see, that's the concern. Also, when you looked at the specifications, the dates and raisins had high allowable yeast and mold specs, right? So Candida is associated with raisins and dried fruits. So maybe you know where it came from, maybe you don't. You don't have all the data in the background, but you certainly have a good idea that says, all right, I could have this, I could have Candida in my raisins. I may be on the borderline for water activity, and now I may create an opportunity for those organisms to grow. So really, if you look at those last ones, it's what can we do with the rice syrup? And there's a couple things to look at. The water activity is obviously the much better on the 60 DE syrup, right? But what I didn't highlight in here is the maltose, right? So if you go 6 rows down in each of these columns, you can see with each successive level of hydrolysis, the level of maltose goes up. Now, if we go back and we look at Malcond's series of cups, where you're looking at the maltose on the left and the sucrose on the right, and you see browning and all the different factors, well, now you have to think about when you do that type of reformulation, what going to this higher DE rice syrup, which has the water activity that you want, what going to that level. One other thing you might have to look at long term is my bar hardening? Is the color changing? What other quality attributes might be at risk as I lower the water activity to a safer level? So just one more thing that comes out of that, those ratios, and something to keep in mind. Obviously, you want to be to the right, but you might change the color or what people are accustomed to seeing. So, the study obviously points to water activity control, and then what can we do for incoming microbial specs, right? So, if you could get it down to 0.65, that's great. It certainly seems reasonable with the lower water activity rice syrup. They use, there's a fair amount of water activity with our water activity control with date paste and raisin paste in each of the formulations. One thing to consider, though, is those are certainly thicker than the rice syrup. And how do you make sure they're uniformly distributed? So you have pockets of date paste or raisin paste, and are they evenly distributed? Is it evenly distributed throughout the bar to make sure you have water activity control, right? Because those sugars are not going to migrate that fast in that matrix, and you may have areas or pockets that allow for growth. So anyway, we talked a little bit about that water activity. So you recommend the new higher brown rice syrup. Obviously gets to it. We need to look at that maltose level and does it change the others? And then what other additional solids can you add? Are there other reducing sugars that will help lower the water activity? Malcond shared that graph. It was just glucose and fructose and different combinations of those, looking at what their impact on water activity is. Fructose, the reducing sugar, was clearly in the safe range. And then on top of that, how do you tighten up your specifications with your suppliers, right? These are ready-to-eat products. What levels can they do for yeast and mold? Can they add the Candida test to the specification to make sure that you minimize that risk from a raw material perspective? And now that if you get down around 0.65, it really gives you a better opportunity to control it without having to get all the way under 0.6, because you're accounting for some of that in the raw material spec. Product's doing great. They're actually growing very nicely. And they've got the problem fixed by managing those different aspects. Excuse me. So as we look at, what type of, what have we reviewed today? What are some of the questions you can ask as you're doing your product development work or you're talking to suppliers? Malcond mentioned it earlier, right? The product design, you got to account for the product quality attributes, right? Do I still meet consumer appeal with the method I'm using? How am I going to make sure, because taste is king, it always is, right? How am I going to make sure I meet those? And the only other way to really ruin a brand is food safety, particularly like if you're a small brand and you have a recall and you're spending $250,000, which a large multinational would kind of say, oh well, big deal. But if that's what it costs at this kind of startup stage because you haven't accounted for these factors, it's a serious risk, right? So when you look at the product quality attributes, right? So what impact does the food safety approach have on that, right? Glycerin versus reducing sugars, right? They both control water activity. Reducing sugars, maybe you got to worry about browning, right? So is there a balance between what I want on the label, what I want the product to look like, and what the consumer segment I'm going after what are they going to look like, right? If you're high protein, obviously you have solubility and hardening issues. Depending on the texture, it could be anything from what types of blends do you use, and all the protein suppliers have blends or unique proteins to help manage this. So obviously sourcing is a key to this. And then again, as you get higher and higher, I mean, as you see like the David bar, which is 150 calories and 30 grams of protein or something crazy like that. You get denser and denser in the protein. How do you make sure that doesn't harden? What type of ingredients do you need? How do you manage it? And as we've talked about throughout this, really just the food safety and the risk factors. What do you need to control? How are you going to control them? How do you make sure you're in that kind of safe space? And if you're not all the way below 0.6, what are you going to do with your raw materials, right? And certainly with the beverages where we talked about the microbial challenge testing, for any of those like multi-use, multi-serve containers, that's always a concern based on what type of application you're working on. So thank you for joining us. I think, Jen, I'm gonna throw it back to you for questions, and we'll go from there, correct?
All right, perfect. Thank you. Thank you both. Yep, so we have a few questions here in the chat. So the first one we have is, what is the risk of having aflatoxins on cold formed bars?
Great, that's a good question. Essentially, like Ed was mentioning, you need to manage the quality of the incoming ingredients. The lower water activity products, such protein bars, will not allow for microbial growth. So the risk of generating aflatoxins in the product is low, since the water activity will not sustain microbial growth. So there's not going to be opportunity for microorganisms to grow and metabolize this, and produce secondary metabolites. So on the product itself is a no. However, if the ingredients are coming contaminated already, the risk still exists.
So that would be more just manage that raw material spec. So if you're bringing in high risk ingredients, you make sure you have the aflatoxin spec testing.
Yes.
Perfect.
Correct.
Thank you.
Good question.
Thank you. Thank you. All right. We have another question about cold formed bars. So if cold formed bars do not sustain microbial growth, how is it possible to find protein bars with probiotics?
Well, that is actually very interesting too. Protein bars can have probiotics in the form of lyophilized cultures or If it's
Spore formers.
The spore
Some of
formers.
The bacillus ones are all spore formers.
right, so they're stable. They don't need to grow in the bar, so they are actually added as an ingredient, as an inclusion at the right dose. So they're still present. They're not active. They're not live microorganisms like you find in dairy products like yogurt or sour cream. So these are not active. However, they still have the probiotic effect once they're ingested. So yes, you can find them. Yes, the protein bar is not gonna spoil because of them, because they're not gonna be growing, and it's feasible. Yes.
All right, awesome. What considerations should I keep in mind when formulating protein bars with inclusions?
So the inclusion, really there's a couple things you need to look at, right? So obviously, if you're doing a high protein bar, the dough gets very thick, very heavy. And when you add those inclusions, now there's a couple different types of inclusions. You could be adding some sort of like nut or raisin, but here if we're talking about puffs and crisps, you really have to consider how late in the mix cycle can I add them? What type of bar forming equipment do I have? And will those crisps make it through the process, right? So if you're using an extrusion process where products tend to be more uniform, you have a lot of pressure, you're going to break those crisps down. If you add the crisps too early in the process or in the mix process, even if it's like say a more of a cold form and you're just using rollers to press it down as opposed to some of the other methods, you're still at risk of breaking it down. So you want to add like crisp, for example, late in the process to help maintain integrity. If you're adding, say, fruit pieces or other things, they really have to come in shelf stable to begin with, right? Because you're not going to get, say, the rate of diffusion or the uniform water activity throughout the product right off the bat. And if they come in with any type of load and aren't safe, then you have other concerns. So on kind of those, so the inclusions, you're really worried about breakage and how you add them to the product for the crisp type inclusions, and then you have to worry about water activity and uniformity for things that might not be well, as uniform, I guess is kind of redundant, but that's what we're talking about.
All right, perfect. And then it looks like we have one more question here. How do you prevent moisture migration on a fruit-filled cold form bar?
Yeah, if I'm
right. So if you're looking like a Nutri-Grain or something like that, right? You're really gonna look at what's the water activity between the two sides, right? So water will typically move from one side to another. I mean, even if you use like a crustable, right? That's kind of a water activity sandwich, right? Like how do you control, what do you need to add to the jam part to make it more like the peanut butter, to make it more like the bread? I mean, peanut butter is not really a great example because it doesn't want to absorb water anyway, but you're still managing, say, that jam and the bread piece for an extruded bar like that. So you'll want those water activities pretty close together so that each piece is microbially stable separately, and that the texture doesn't change by, say, the coating having a lower water activity and trying to pull all the moisture out of the filling, for example, right? So you'll want them to be as close as possible when you formulate it in that example.
All right, awesome. Well, that looks like what we have for the questions. So I just want to say thank you both to you gentlemen for speaking today, and just want to say thank you to everyone who's also attended. Please definitely feel free to reach out with any questions. We would be happy to connect further to discuss this important topic. This now will conclude today's presentation. Have a wonderful day.
Thank you, Jen.
Yeah.
Thanks, everybody.